The Tag Futile Cycle by Jaime Coverti. If fatty acid synthesis and beta oxidation were to proceed simultaneously, the two processes would constitute a futile cycle, wasting energy. A futile cycle, also known as a substrate cycle, occurs when two metabolic pathways run simultaneously in opposite directions and have whatsoever no effect other than to dissipate energy in the form of heat. The reason this cycle was called the futile cycle was because it appeared that the cycle operated with no net utility for the organism. After further investigation, it was seen that futile cycles are very important for regulating the concentrations of metabolites. For example, if glycolysis and gluconeogenesis were to be active at the same time, glucose would be converted to pyruvate by glycolysis and then converted back to glucose by gluconeogenesis, with an overall consumption of ATP. Futile cycles may have a role in metabolic regulation, where a futile cycle would be a system oscillating between two states and very sensitive to small changes in the activity of any of the enzymes involved. However, with beta oxidation, as read previously, is blocked by malenyl-CoA, which inhibits carnitine acetyltransferase 1. During fatty acid synthesis, production of the first intermediate malenyl-CoA shuts down beta oxidation at the level of a transport system in the inner mitochondrial membrane. This control mechanism illustrates another advantage of segregating synthetic and degradative pathways in different cellular compartments. A study done by the University of Pennsylvania Medical Center in 2002 asked the question, if obesity is the leading cause of type 2 diabetes, then why do TZDs, which is the only available drug that treats this form of diabetes, actually cause more weight gain? TZDs from research describes how they trigger the creation of glycerol kinase, an enzyme that causes fat cells to store fatty acids faster than it produces them. Researchers call it a futile cycle. Just as fat cells release their lard or fatty acids, glycerol kinase causes the fat cells to put them back into storage. This is quoting Mitchell A. Lazar, MD, PhD, Chief of the Division of Endocrinology. The glycerol kinase, as said by Mitchell, is packing the pantry faster than fat cells can pull the cans off the shelves. Basically, the net influx of fatty acids into fat tissue contributes to reduced fatty acids in the bloodstream, which in turn lead to increased sensitivity to insulin. TZDs actually lower insulin requirements in diabetics, and this is likely to be beneficial in terms of the risk of hypertension and heart disease associated with diabetes. To help understand this process, I've put a picture called the fat feeling cycle to help explain in better detail the process. When glucose is high in the system, insulin must be expressed to lower glucose levels and help with glucose control. Fatty acids are then made and want to be broken down through the beta oxidation and citric acid cycle. However, due to glycerol kinase and blocked CPT1, due to malenyl-CoA in the futile cycle, fats will be stored first and can lead to weight gain. The TZDs mentioned in the study act as insulin in this futile cycle. Okay, now quiz time. Yes, I know. Get your eye clickers ready. You have 30 seconds. Malenyl-CoA is a highly regulated molecule in fatty acid synthesis. It inhibits the rate-limiting step in beta oxidation of fatty acids. Malenyl-CoA inhibits fatty acids from associating with carnitine by regulating the enzyme carnitine acetyltransferase. It then prevents them from entering the mitochondria, where fatty acid oxidation and degradation occur. The release of insulin triggers a series of steps that result in the activation of acetyl-CoA carboxylase. Release of insulin indicates high food levels. Glucagon and epinephrine, both hormones, inhibit the enzyme through a series of steps. In mammals, the enzymes necessary to synthesize palmitic acid from acetyl-CoA and malenyl-CoA are present in a complex known as fatty acid synthase. In plants and bacteria, the enzymes are present as separate molecules. Synthesis proceeds 2 carbon atoms at a time, which is why all natural fatty acids contain an even number of carbon atoms.